1 second. Maybe try the clicker now. Here we go. Yep. Okay, I think we are all set up. Are you all good? Do you think so? Okay. I will welcome everyone to this year's Myra Samuels Memorial Lecture. This lecture is named in memory of Myra Samuels, who was an associate professor of biostatistics and epidemiology at Purdue University's Department of Veterinary Pathobiology, and associate director of statistical consulting in the Department of Statistics. She taught at Purdue for 24 years, and her research was focused on biostatistics, conceptual issues in mathematical statistics, and collaborations on applications. And this year's speaker is Bhramar Mukherjee, who is the John Kalbfleisch Collegiate Professor and Chair of Biostatistics at University of Michigan, and is also Professor of Epidemiology and Global Public Health, as well as the Associate Director for Quantitative Data Sciences. Her research focuses on statistical methods for electronic health records, studies of gene-environment interaction, shrinkage and estimation, and analysis of high-dimensional exposure data. She's co-authored more than 300 articles in the fields of statistics, biostatistics, medicine, and public health. And she's received numerous awards, including the Gertrude Cox Award from Washington Statistical Society, the Adrian Couples Award from Boston University, and the Distinguished Women's Scholar Award from Purdue University. And today she'll be talking about using public health records or electronic health records for scientific research. Let's all welcome her and give her. Oh, before we get started, Ben's going to present the award.
Those of you on Zoom cannot see that.
Those of you who are on YouTube and Zoom, Dennis is actually presenting the award. So if cameras.
I think the camera's on the wrong one.
So
okay, this webcam is working now.
Oh, good. Wow,
excellent.
Maybe slide out of the projector.
That's projector.
Yeah.
How about that? Okay, don't lose it. All right, thanks everyone. Those of you who are in person, special thanks to you. Those of you who are on Zoom and YouTube, you are missing a big event in person. So I first want to thank the Department of Statistics at Purdue University for establishing this award. And I just want to present some of my research in electronic health records today. For the last 2 years, as many of you know, I have been deeply involved in modeling the pandemic in India, so I have not gotten to much of my methodological research, what I'm paid to do. So it's great to be able to talk about those methodological issues, which has been occupying me for the last 10 years or so. But before I begin, I really want to thank, the advancer.
I think if we click on the screen, if you try the clicker now, it should work.
So I really want to thank the previous speakers. The previous honorees of this award. This really shows, so this is the 27th lecture, and we have honored 26 outstanding statisticians in the past. And as an alum of the department, I feel incredibly proud of the legacy that Purdue Statistics has established. So I want to hear a big round of applause for all of this, our profession in general, statistics, biostatistics, probability, all of them together. So I wanted to, Purdue, I wanted to share my tribute for Myra. And this lecture was actually established in 1996, my first year as a graduate student in Purdue. So this really shows you the full circle of life or academic life. Many of you who are students listening to the lecture, one day you will be presenting. So this is just spectacular things that happen in academics, which you never think that will happen. So Myra, when I was in Purdue, I really found in every office this textbook, Statistics for Life Sciences. And as you can see, she wrote it in 1989. Even at the time, there are a lot of introductory statistics textbooks, but introductory biostatistics textbooks with a lot of applications. And because she worked in the consulting division and also was a professor of biostatistics and epidemiology in the veterinary pathobiology department here, she had access to lots of ongoing interesting research problems. And so if you think about the 1990s, those were like Purdue was a very theoretical department at the time. And our field in general has evolved a lot. And Myra was sort of a pioneering statistician who could actually see the field. And you can see that this book is on its 5th edition, right? So in 2012, so several other people joined to revise the book. And now in the 3rd edition, it became Samuels and Whitmer, and then it became Samuels, Whitmer, and Schaffner. Myra passed away in 1992. 20 years later, the 5th edition is being revived by 3 statisticians. So I really, really appreciate this continuity of fraternity and friendship and collegiality in our profession more than anything else. And being in Purdue reminds me of this. And you guys have a wonderful resource, which we do not have. And I'm going to borrow this idea for Michigan Biostatistics as the chair, that you have this wonderful archive of old photos actually it's publicly searchable from the department website. And I tried to find photos of Myra in the Christmas party. And as you can see, Myra and Steve Samuels, her husband, who was my professor when I was a student in Purdue, are singing. And I think that this is Herman Rubin on the piano or accordion or something, some musical instrument. And this was just amazing. Okay. So I actually, this is wonderful to go back in time and see that how fun the department was and how fun the department still is. When I was a student, this little garden was established in honor of Myra. And you can see the little flowers blossoming. She left us way too soon, but her legacy and her memory really lives on. I still remember, this will really reveal my age, but I'm not afraid of being old. So this is the first Myra Samuels lecture and the dinner in his honor. In 1996, this was my first semester as a graduate student. As you know, that if you come from India, you're getting used to the weather, you're getting used to the culture, you're getting used to the food. But I still remember that the first person who presented the lecture, Herbert Hauptman, was a Nobel laureate. And we were so excited that a Nobel laureate is coming to a department of statistics. And so this is something that I treasure and remember. And what was I doing then? I just want to, because this is my home ground, this has been a wonderful homecoming. As I was driving yesterday, I actually cried to see Wabash, because it has so many fond memories. My daughter was born here, so Purdue is just a wonderful place to come back to. So this is what I was doing. This is we used to have coffee with the math department here. So of course, as you know, that mathematicians do not think much of statisticians, but we still managed to get coffee and pie together. And we were really planning to change the mathematical universe over coffee and pie. We were talking about all the $1 million problems that we're going to solve and become rich. So I'm not going to report on our progress there, but this is our cohort, and they have been such wonderful friends through life and such smart people. My life as a graduate student was very different because I was a graduate student parent. I took my PhD qualifiers 10 days after my daughter was born. Tom Selke was the proctor, and I can still remember the look on his face when I showed up for the exam just after having a baby. So, but it is a different philosophy of life, right? You are a parent, so there are more important things. Your daughter is sneezing. It's more important that you cannot prove a theorem. You work all day, and this is honestly the blackboard in our office in the 7th floor, where me and my advisor, Professor Studden, will try to prove very important theorems all day. And then, even before I captured it, because there's no cell phone, you couldn't take photos. Even before I could jot it down, all these complex ideas, my daughter, shown in this picture, she was 3 years old, will come and paint a picture of a flower. And all this work is lost, but there's something beautiful about it. So she always was raised by, when you were poor and you were a graduate student, you actually get a lot of support from your fellow students. When you are a faculty, that suddenly disappears. You are on the other side, that you're supposed to take care of yourself. But graduate students have this wonderful solidarity, and you can see when Chisai and Nitai Mukherjee, 2 of my cohorts, are taking care of my daughter. She fell down, and you can see that she had 9 stitches, and she would not stay with anybody without these 2 people because they were her favorite. And they also were very smart babysitters. And for the longest time, my daughter did not know that you can stop at a bachelor's or a master's degree. Her thinking was that you can only apply for jobs after you finish your PhD, because she did not see anybody else. So that was a good brainwashing period. But then my thesis, right? So what I'm going to talk about today is probably in any norm very far from what I did for my dissertation. So this is my advisor, Professor Bill Studden. He's not with us anymore, but I really appreciated Professor Studden's strictness and his standards. So this is what you can see, what statisticians in the biostatistics department call pre-post experiment. And that experiment was having me as a graduate student. So this is him before and after I finished my PhD. I was his last student, not to mention. I was one of his 3 Facebook friends. He is no longer with us, and I still remember him very, very fondly. And after years of hardness, I think that, what I remember from Professor Studden is his standard, his rigor. But I think that he also gave me a part of his invaluable collection of math and stat books, which are in my office. And so this legacy goes on. That's sort of a theme of my talk, that we are all connected in some way. But he did not understand me. But parents often don't understand their children, right? So what I wanted to do and what he wanted me to do were really miles apart. So how do you reconcile? I think he always asked in his head, how did she end up in biostatistics? If you look at his CV, 80% of his papers are in Annals of Statistics. He did not consider any other journal as journal. I still remember when I called him after getting tenured at University of Michigan, I said, Professor Studden, I got tenure. Guess what he said? 1 syllable. How? So he was very tough parent, right? I know that he was very happy for me, but he could not think in his spectrum that, did I manage to sneak in a paper in the annals to get tenured or not? That was the culture. So we have come a long way. Now I'm the chair of a biostatistics department, and many of my faculty get promoted without publishing in the annals. And I'm not saying that should be the norm, but I feel like we are broad enough to embrace all kinds of statistics. So it was very important for me to see very strong women role models at Purdue. And I just want to give a shout out to Mary Ellen and Rebecca for really holding my hand through a day when I was not sure that I would be anything. I could make anything of myself, struggling as a poor graduate student with a thick accent and with a little young child crying all the time. That was miserable. But without the help and support of Purdue fraternity, which is like my family, I would not be anywhere. So till this day, my friends at JSMs and everywhere, I could sneak in a picture of Doug, who has been like really a constant figure in my life because he debugged all my Mathematica code, because Professor Studden would not touch Mathematica. So you can see that our cohort still gets together. So maybe in a day-to-day life of a graduate student, you always think that, oh, what I'm proving, my papers, my exams are the most important thing. It's really not. It's really the people. It's really your friends. It's really the faculty who surround you who are going to be there for the rest of your life. You're going to forget all the bad grades that you got. You're going to forget like all the theorems that you could not prove, but the friends will be with you. So with that, I think that's my first like tribute. I want to talk about something research. Otherwise, Professor Studden will not be happy. So I want to, my father is an actor and director in India. So he always told me that you statisticians just cram too much material. You do not give people any chance to absorb what you're saying. Divide your talk into 3 acts. And after each act, give the option to people to leave. Because they're almost buying a ticket to see you perform. They're investing that time. So they can either boo you or clap for you, but give them a pause, give them an option. So I would not quite give you that option, but I would like to divide my talk into 3 acts where I'll pause after each act for us to go through some of the salient points and for you to ask me questions if you choose to. Because sometimes we talk about so many things that it's hard to keep track of things at the end of the talk. So that's what the plan is going to be. The first act, I want to talk about the data-rich environment that we have created at University of Michigan. When I was in Purdue, and this is the challenge of every statistics department where there is not a strong medical school or a school of public health, is to get access to very interesting data, non-trivial data sets, which are going to raise interesting questions and on which you can write your grants, on which you can write your papers. Your ideas come from the data, not that you prove a theorem and then look out for where can I get the data set to illustrate my methodology. It's the reverse process, which I find absolutely scintillating. So I want to talk about how we built that and how, as a statistician, I contributed to that process and also rolled out my first epidemiology cohort study. So that's something that I want to share with you because this is unusual for a statistician to be PI or be leading and creating a study. And then I want to talk about the methodological meat that, yes, there is data, but there's very imperfect data, and how can we use our statistical skill sets and our toolboxes to make inference better. The data we have, you can never model your way out of bad data, but you can be less imperfect. And so that's what I'm going to show you. And then I want to give you a glimpse that because of Act 1 and Act 2, because of 10 years of work, when COVID happened, we were actually very nimbly positioned to do some research. Without the previous work, in 2 years, we'd not be able to do the research that we actually were able to do. All right. So before I begin, I want to thank 3 individuals who contributed to the EHR research incredibly. Lars Fritzsche, who is a human geneticist, Lauren Beasley, who is my statistical postdoc, and Max Salvatore, who is an epidemiology PhD student. And the reason I'm showing you this 3 different kinds of people is that you need a team. Only statisticians cannot really tackle very big, massive data problems because you do need other expertise to come together. And we are fortunate to have such an incredible team to think through these problems. So in terms of methodology, the reference that I'll share with you, and these talks, I shared the Google Drive link with men, and they are publicly available. So this paper just got published in 2022, but this is really the key methodological paper that I'd refer to. So University of Michigan started this precision health initiative about 2012, 2013. It's a $100 million initiative where the investment came from the provost's office. And the goal was to create data resources, just integrate different kinds of data on a lot of people. So we are thinking large N and large P, which will serve as an accelerator and incubator for interdisciplinary health related research. And it is hypothesis agnostic in a sense that you could easily validate your existing hypothesis, but also you can mine the data for new ideas and new hypotheses as well. So, and the goal was that this return on investment will bring back many other grants, many other methodology back to campus, and many people, many faculty will be able to use it. So the way it started was collection of biosample or blood to get DNA genotyping from patients and get their permission to connect the genetics to their electronic health records. What we wanted to do is to really create a cohort, and it started in 2012, but the sampling was really very forward thinking. 10 years ago, we actually had a one-page consent where we not only got permission to connect electronic health record to genetics, but also to any other national level or any other record that can be linked in a confidential manner, confidentiality, privacy preserving manner with this record. So that means that through social security number, we could actually connect other fields of data. So that was very bold thinking. And we could also recontact the patients if we find something, right? So that's also important, or if there is a future study. So there are many, many biobanks. Many people are using the UK biobank, which is about 500,000 people with many, many different types of data, including genetics and imaging. But I really care about the Michigan study. I care about the Michigan Genomics Initiative because there is a real potential to actually transform health if we find something to go back to the patients. And I often hold community participant engagement studio with the participants of this study, and they tell us what they want from researchers in Michigan for improving their health and the health of a community. So on this picture, I care about this study more than other larger, bigger, better studies because I know the physician, I know the patient, and I feel like we can make a difference at Michigan. So MGI is very big data because you have about 21 million genetic variants after imputation. And medical encounters, as you know, that every time you see a physician, you get some disease codes or ICD codes, and that generates, and you get lab tests done. And so that's a very rich data set with very big dimension as well. And so we have about 80,000 people now. We are actually genotyped, but 100,000 will be soon the milestone for this study. But we all agree that a genetics and electronic health record does not really codify a person's individual's existence. There's a lot more: behavior, lifestyle, whether I took the stairs to come up to this room or not, family history of cancer, environmental factors, social factors, which we're able to collect and generate. So as I said, I was very unhappy with just the EHR. And the EHR was the patients were recruited actually in a very biased mechanism prior to undergoing surgery, right? So they were all waiting for surgery when they were recruited in the anesthesia clinic. So not everybody in the population goes through surgery. So obviously you'll see different kinds of enrichment motivating the methodological work. So we wanted to really adjust for confounders, right? Because we have a bias sample, we have confounding issues. So we collected a lot of behavioral and occupational exposure variable. And this was my study. We have 7,000 people in it from the Michigan Genomics Initiative. And the idea was that if I'm interested in cancer, everybody will have this top part of the data. But if I am interested in cancer, I can deep dive and have a cancer questionnaire. If somebody is interested in mental health, they can have a deep mental health questionnaire. And then somebody is interested in perioperative outcomes, they could have their own questionnaire. But everybody will have access to all of these because we don't really want to measure this over and over again for each individual. We want to have a common frame. But then what happened, like with all studies, the main study was this 70,000 in surgery. But then there were so many other clinics who started sampling. Some studies came from pediatrics, some came from mental health. And so everybody followed a different kind of sampling mechanism. And as a result, what happened is that you have a hodgepodge of different individuals. So you have a lot of people, 90,000 people, enough to estimate a parameter of interest, but not from a well-defined probabilistic sampling frame. And any computer scientist who tries to grab this data and fit a machine learning algorithm should be aware of how data are made or how sampling is done. So that's sort of my pet peeve in terms of electronic health record data that we do. Yes.
We have a question from the chat. Is the
Sure.
MGI data set available to the public?
Yes, it is. And I have a last slide where you can actually request the data. Yep. Yeah, so that's one reason I'm actually showing this, because there are many people here who are probably looking for rich data sets to try their methods on, or a source of problems, and MGI is available. So what we have been able to do is we have linked the core data element, the genomics, the epidemiologic data, with other registries. For example, we have very good quality death data now, because people interested in survival analysis really complained about the poor quality of survival data in EHR. So we connected that to cancer registry, we connected that to national health registry. But also one thing which really helped us during COVID was we had mapped the geocoded residence information into census tract level variables in terms of socioeconomic status, neighborhood quality, crime rate, pollution, many other risk factors at the ecological residential level. So we had a lot of neighborhood characteristics mapped already. And then we also connected that to medical claims, right? And the dental records. So all of these, the different data sources were siloed, right? It will take you years to put them together. If an university takes the effort to put all of this together in a usable format, that will be real accelerator for your research for what you want to do. So when COVID came, we already had this. That was our biggest advantage. Then we actually could take the COVID vaccination data, COVID outcome data. Now I'm doing a lot of projects on long COVID and COVID symptom survey. I'll show you that as soon as COVID happened, we could actually survey our patient population to see how they were feeling about COVID. And we have continued that effort. So having an engaged patient population was very, very important. And during COVID, how we actually collected data is we mailed them saliva kits so that they can, these are spit kits that are being sent. We did not stop. We still collected biosample. We still wanted to do genetics. And it turned out for epidemiologic study, the last 2 years, particularly the first year of lockdown, was phenomenal, because we were bored out of our mind with our playing with our phones. So it was a very good time to do surveys. And so I could not believe that actually we have about 90% completion rate on all of the surveys that I showed you. So this obviously tells you that probably not a representative sample, but very good response rate. So we did not stop. And we also added surveys, as I said, in the early days, how Michigan residents are feeling, right? So that's why it stands out for the state, for the university, that we are focused on our catchment area. We have similar types of surveys all over the world, including Facebook surveys. But then what were they feeling about? What were the perceived risk of severe COVID-19 and what were some of the risk factors? This was very early days of the pandemic in June that we were able to survey our patient population. And then I was really interested in who is getting tested. People are getting tested all the time. In the first period of testing, only few criteria will enable you, make you eligible for testing. If you had symptoms, if you had certain comorbidities, but who is getting tested repeated times? And then who is actually getting the antibody test, right? Many of us are concerned, do I still have antibody? Is my immunity waning? So who are these people who are getting repeatedly tested? Because unless you have a model for selection into your study or who is in your data or who is being diagnosed with COVID, you'll not be able to do inference. So we published in medical journals many papers trying to draw your attention that people need to pay attention to who is in the sample. Whose data are you seeing? Because there is a huge amount of people whose data you are not seeing if you condition on who got tested. So, but it was very exciting to actually have electronic health record data during this time where we were getting a data dump every month. So I'm just showing you, this is for my personal interest. I was just paranoid. Are the vaccines working? Are we having antibodies? So we were looking at the antibody test, the IgG test throughout time, so this whole time from 2020. And you can see that this is what percentage was positive. This is just through infection, right? And so this is sort of giving you a prevalence of how the disease, mostly it was negative. And as soon as vaccination started, this rose, right? So of course you can say that this is wrong, right? The prevalence is wrong. I'm absolutely sure the percentage of positive that you cannot really estimate from this data, because again, it's the bias who is getting tested, but you can definitely see something is going on. And that is something I'd like to reiterate throughout this talk, that there are certain things you cannot do, but still this data contains information, because this is real time what is happening, and it's very actionable how medical system is working, right? So that's very interesting to me. So the Act 1 is coming to an end. What is our vision for this? I showed you that being involved in a process like this as a statistician, I think the vision is that we have a rich data set with large N and large P, where many investigators at Michigan and outside Michigan can explore through different lenses. If you are interested in socioeconomic status type variables, you are interested in drugs and prescriptions, you can use it in a hypothesis-driven way or very agnostic way too. And our questionnaires are very similar to the other major biobanks in the world. So if they are doing a meta-analysis of genetics versus one question, we can also contribute to that. And this has really accelerated health research in Michigan, because if you have that broad database, it's much easier to tell the funder, we already have this, I just need metabolomics data on 5,000 people, right? Because I can connect to all of these other things. So, and then during COVID, we saw that how we can pivot very nimbly to address some of the emerging challenges. And we were working with the physicians who are taking care of patients in the ER to really, I'll show you one example in the end, that who is at high risk of COVID-related complications, because not much data was available at that time. Many papers have come out since then. So that's Act 1. I'll pause and take any questions that you may have before I go to Act 2 and talk about some of the modeling work that we did. Yes.
So when you build this integrated data source, do you have large meetings with people from different departments every month or every 2 weeks?
So the question is that when you build such a resource like this, do you have meetings, large meetings with multiple departments? So yes, meetings are a part of life for building anything. I always say that chairs are always at the table, right? So poor joke, but you really do not get to do much research work during the day because you're always at meetings. But Michigan had a really organized infrastructure where each school actually chose representatives. And so I was the associate director for cohort development, and I deliberately, intentionally actually chose to serve in that role because sampling and who gets recruited was more important to me. I was not happy with the surgery sampling, so I wanted to really broaden. I wanted to recruit underrepresented minorities on whom we do not have good data. So I think that there is a data analysis course. So we're a very interesting infrastructure where every group is leading every component. So there's the data, and then there's this whole data and analytics group who can share the data in a privacy preserving way with the rest of the campus. So there's a huge team working on that too. So this was compartmentalized into different groups, and each area had its own leadership. So, and we had a national advisory board as well. Yeah, it took us 10 years, but it was lots and lots of meetings, yes. And privacy and confidentiality is a very big issue, right? Because you are connecting to so many things, neighborhood level information. You could not do it for rare diseases and so on. So there was like really a lot of nitty-gritty here. Yeah.
So even since they're supervised, there's a security or confidentiality team that supervise in data.
Yes. Yes, yes. So the medical school has a research data warehouse because this medical patient level data is a real big deal, confidentiality and privacy and security. And so they have a very big team expert in this. They generate encrypted IDs for each user so that they sort of work as an honest broker. None of us can link the data without their help. And each investigative team has a set of different encrypted IDs so that we cannot share with each other as well. Yeah, a lot of thinking has to go in. Yes.
So people agree to be in your study, they have to agree to link to other databases in that way.
Yes.
Do you think that introduces a big bias in the kinds of people?
I will show you I thought like you. And then I was surprised by the data. I'll show you some of those because that's very important. Yes.
Kind of following up on that same idea, at the time that they agreed to be linked up, things have changed dramatically in terms of what they can be linked up to. So did you have to go back and reconsent or do anything like that?
That is such a good question. So what happened was so this is one of the main criticisms of the study. I consented in the study without even probably recognizing the full spectrum of what is expected because I was waiting for my surgery, anxious. Somebody came and talked to me, I just signed, right? So we actually have to revisit and call them every 5 years to say, and I would tell you many of them forgot, but that they enrolled in the study. But when we remind them and tell them you have an option, they have such trust in University of Michigan researchers. They say, I'd give you anything to help other patients. And that's the same thing you hear over and over again. And the consent rate of people we approach is 80%. So that, and even when you remind them they want to stay, very few small percentage actually drop out. That shows people, and when you hold this participant engagement studio, most of the time it's, Michigan health system has done so much for me, or some of my family members or friends. If you can improve cancer, we'll give, if my data could be of help, we'll give you anything. Yeah.
So,
yes.
in addition to the EHR data, so what are the omics data available?
Yeah. So Min's question is that in addition to EHR, what is the omics data available? So let me go back to the core elements is EHR and germline genetics, right? Just GWAS data. But then different people have gathered different data. So we do not have exome sequencing. We do not have proteomics. We don't have metabolomics or microbiome data on everybody, but we have on different investigators and maybe 10,000 people out of the 100,000. So that's the goal, that gradually people are going to get grants so that we can have more, like now it's a pattern sort of missing data, and we would be able to fill in as people get grants to say, just collect microbiome data on every GI cancer. Things like that are happening. And one condition for using all of the other data is that you have to let your data after a couple of years to be used by others. Yes, Doug.
So do you take steps yourselves to clean this data as you put it in the biobank, or does everybody else just have to clean it themselves?
That's a great question that Doug is asking, that our team have to clean the data? So for the genetics data, it goes through very rigorous quality control so that we have very standardized genetics data. So there is a pipeline for that. For electronic health records, the clinical notes, because that's where you have the highest risk of revealing people's identity. They are high priority and they are not given to everyone. You really need very high level access to get the clinical notes. The structured data, lab values and so on, there are processes that's actually cleaned. And so we get pretty clean data, I would say, because we want to have a standardized data harmonization across all investigators. But some data cleaning have to be done, or decisions have to be made that what are you going to use in your analysis? Yes. But the whole centralized team doing it really takes the pressure off of individual investigators. And many times, in biostatistics department, we know a lot about data, but someone from pharmacy wanting to use it, they will need a lot of help, right? So the centralized infrastructure is very agile in providing that support. Other questions? Okay. Any questions on Zoom?
Right now.
Okay. All right. Thank you. And so electronic health records data. And I have been working on this for the last 7 years, and it has evolved a lot. It used to be very poor quality because when clinicians were first asked to put in the data, they were very upset. When just think about the process when you're visiting a doctor, along with seeing you, they have to input all of these disease codes that they are supposed to be used for billing. That's not really satisfying to them. It's mostly used for billing, and that's why hospital administrators love it. More code, more payment from the insurance company. Recently, I realized that malpractice lawyers really like electronic health record because if something happens to you, they can go back and say that at 5:37 you're supposed to get this medicine and you didn't, and so on. So they really do this. This is new to me. And then of course, data scientists and informaticians really like us like it too. But what is really the pro is that in 10 years we are able to collect all the data, connect all of them. And we have health history, right? We have longitudinal data. So when COVID happened, we already knew in our patient EHR, particularly for people who seek their primary care at Michigan, what were their existing comorbidities, right? Do they have diabetes? Do they have hypertension? What puts them at higher risk for COVID-related severities and so on? The cons are that you do not, you have a control, like, you don't have an experimental design. You do not know why a patient is there. That's the fundamental framework that we need in order to do inference, that what is my sampling frame? And some of the measures are very, very poor. Generalizability, transportability, representativeness, some of the classic concepts in epidemiology are very much needed, which are often being ignored by the jazz of the sample sizes and all of this data that we have. So I'm going to be I championed in Act 1, this act of gathering data. I'm going to bring it down a little bit in Act 2 by saying that, so what? So, and so you have, but this is not an usual, not just a general, like, MGI or Michigan Genomics Initiative problem. We are seeing people taking lots of data from lots of sources and using it for scientific research. These databases were never really designed for subpopulation-based research. They are designed for other purposes. And in my courses at Purdue, Anirban always taught me about relative efficiency. Focus was very much on power and efficiency in the classical statistics that we learned. But in this big data, it is becoming more and more clear that you have large sample size. If you are interested in one or two things, you're okay. But you really have to think about bias, because the data is collected in such a way that the bias is not going to go away with sample size. So that's something which is really, really important. And I borrowed this slide from my colleague, Rod Little, where he shows this very toy example that suppose you are targeting to estimate true proportion. Suppose this is true value is 0.4. We are very used to well-characterized studies, randomized clinical trials, where we could not recruit so many people, but we don't really worry about bias because we put in so much attention to inclusion and exclusion criteria, have been very careful. So we are sort of in this situation where I know that true parameter is probably within the credible interval if we did things right, but the credible interval is wide, so I don't have that much information. Now I have this big non-probabilistic samples where my sample size is so large that my credible interval is tiny. You'll see for all estimates. So it's very paradoxical in a way, and this has been discussed in the literature quite a lot, that this additional enhanced precision is almost hurting you, because you do have tiny little biases are not being overcome, right? So you had a chance in the first case, even if you are biased, the precision is sort of giving you a leeway of the parameter to be there. But now you have say 0.01 bias, but your standard error is like 0.00001. So even tiny biases are making your inference or the final conclusion incorrect in the parameter is outside that credible interval. So in a sense, you're becoming precisely wrong. So a statistician will probably like to see the information content of the second study and the properties of the first study in terms of its rigor and carefulness to come up with an estimator which does a bias variance trade-off. So I really think that this fact and you know, Xiaolin Meng has written a lot about this in terms of big data paradox. There is a wonderful paper in Annals of Applied Statistics in 2018 about the presidential election, how this type of biased data gave incorrect conclusion. So we are introducing so many courses about machine learning, big data computing. I have this health data science concentration where students are learning how to do Hadoop and MapReduce and so on. I really think that the computer science department should reciprocate. The computer science department should have a sampling course and a study design course, because somewhere basic fundamental mistakes are being made many times, and asking this question that who is in my study and what is the target population of inference is being missed. So without thinking about this, you can accumulate big data, but you cannot translate it to knowledge. And more importantly, you cannot translate it to actionable knowledge. It's not enough for me to just be able to predict. When I signed up for a job in School of Public Health, it's my job, it's my mission to improve health. For that, I need to intervene and know the causal mechanism, why this is happening. Just prediction does not do it for me. So as I said, that MGI is big data, but it followed a very convenient sampling frame. And so this is going back to your question, 75 to 80% actually of the people that we approached consented. So this was startling to me, that people are willing to, because we hear so much about privacy concerns, that people are willing to contribute their research data for academic research. But if we look at the data, about 50% of people actually have cancer in this data. So obviously there is an enrichment with respect to diseases, and it's very obvious, having a role in the cancer center for a long time, I know that cancer patients undergo surgery or interventional procedures more frequently than the general population. So surgery indirectly related to cancer, but some cancer are underrepresented, like blood cancer. Liquid cancers, they do not go through surgery, so that's underrepresented. So all kinds of issues if you even try to estimate prevalence of cancer from this data. So this data was actually collected by my former chair, Gonzalo Abigasis, who is a rock star scientist. And I gave him very, very hard time that you geneticists, you do not care about sampling, you're collecting garbage data, and I'm going to bet 2% of my salary, or maybe like 0.5, that this data is garbage and it is not going to give you any meaningful information. And he just said, Bhramar, why don't you take a look at the data? And I very reluctantly, he was my chair and he's very smart, so I listened to him and started to look at the genetic data. And we published many high-profile paper on doing genetics in using this biobank. And we also created a polygenic risk score, or a genetic risk score repository. But what really startled me as a statistician is when I looked at, on the x-axis, I have plotted, in genetics, you have the advantage, there have been already many, many large studies done. So you sort of have a calibration gold standard. So I plotted the log odds ratio from genome-wide association studies, which are result of multiple years and millions of dollars. And on the y-axis, we have plotted the genetic odds ratios from our NGI, from our own cottage industry study. And this is something which was startling to me. I could actually accept that the p-values or the ranking of the genetic odds ratios will be sort of same in the two studies, could be, but point estimate. But it just tells me that if you have a subselect section of the population and it's such a haphazard sample, there has to be bias in the point estimate. But the point estimates were relatively close as well for many cancers. And as you can see, the concordance coefficient between the two estimates is around 0.9. So why would I do this as opposed to do this? And is this general? So this really let us go to the mission. And I'm going to have to really breeze through these methods because I'm never going to go to the last part of my talk then. That why? Why is this? Why is that picture like that? Why doesn't sampling matter for GWAS studies? Why are geneticists so lucky that they can ignore some of these foundational things? And so I really wanted to know the answer. And the second thing which has always bothered me, that US is not like UK, right? Because we don't have a national health services, so every EHR is connected. We only see patients when they are in Michigan, and we assume that nothing is happening in between the time. So what is happening between these times? There is an incredible missing data problem. And whenever we are seeing the patients, that's extremely informative, right? Most of the time, it's not a wellness visit. You're going to the doctor because you have a concern. And all the lab tests that you're seeing are completely the observation process cannot be ignored. It has to be modeled. And so we are not capturing all of the diseases, or in statistical words, that will mean a less than perfect sensitivity. How do we actually think about the two things together? And when you think about inference, we really have to you have to tell me, what is your target population? Is it the people who you approach for consent? Is it Michigan Medicine, or is it United States or the state of Michigan? And how can I go from my Michigan Genomics Initiative to all of this population? You have to have a framework for that. Otherwise, I'm not just going to review your paper. So how do you deal with selection bias, and how do you deal with this big non-probabilistic sample is something that we tried to really model and formulate. So I want to argue that these two things really matter, and these often really distort our previous statistical conclusion. So I'm showing you a very obvious association between cancer and gender. It's actually very well characterized in the literature that women are at lower risk of getting cancer as an overall phenotype. And so this is the true estimate on the left-hand side. And then artificially, I selected subsamples and introduced misclassification of the cancer phenotype. And we have this notion that if you have measurement error, the estimates get attenuated, right? We just lose power. But that's not true when your sampling is also dependent on what you are interested in. You can, when you have misclassification plus selection, which depends on what you are interested in, and that's the key, that if you are interested in, if your sampling or selection depends on what you are interested in, then all bets are off. You can see that men are at much lower risk, or women are at much higher risk of getting cancer. So that's completely wrong inference that you can get if your misclassification and selection processes are related to what you are interested in. And so I'll give you the answer for the genetic studies. That's where they are saved, because that's the arrow. You rarely select or consent into a study based on the value of a single nucleotide polymorphism on your genome. That association of the selection or the misclassification with the genetics is very weak. And you can prove that. This is sort of really that causal inference confounding missing data type argument, that as long as that is weak, then you are actually saved in terms of bias. But I just want to go through some of the formulation before I go to the COVID examples, that how we model this is really sit down and think what happened for the study, right? So we are interested in a model for D with some risk factor Z, D conditional on Z. And then we do not have the actual D. We have a D star or the EHR measured variable. That's a classic misclassification problem. And then coupled with that, we have the sampling problem, right? That we are only seeing people who are sampled and with the contaminated disease process. So we have to model two data anomalies. One is in terms of observation, one is in terms of sampling. So now we're going to really start modeling this in terms of other variables that you see in the EHR. That's the challenge, that we don't have gold standards for sensitivity, specificity when you are running through the whole EHR. You have covariates which may be informing each of these processes. Why did you get sampled and why did I miss a diagnosis? Yes.
So we have another class meeting at 11:30.
Okay.
So if we could try to wrap up.
Sure. And so, so what happens is that, you can, so basically I'm going to just show you that once you posit these models together for sensitivity, for selection, for specificity, you can actually find out variables in the EHR which are informing these processes. And so the key statistical issue was then, how do you go from the model that you fit to your target model? And that's where something beautiful happens, that you can actually prove a theorem which relates the two model, the target model and the target model and the model that you are after. And so this is the model that you can fit on the selected data with the coarse variable, but then the sampling and the error variable comes in as a very nice correction factor, and you can fit a nonlinear model. It's not in the transform logistic model, which is not in the GLM family anymore, but you can try to do things with this model. And with this model, you can then incorporate how do you estimate this sensitivity parameter, sampling parameter, and you can go with that. And you can extend this result to imperfect specificity as well. But when things sort of follow into places, they're clean. Professor Studden taught me, like whenever I went to him with the proof, he will say, this is correct, but this is a proof. This is not the proof. So when the proof happens, so things look simple. That, as you can see from your case control study knowledge, the sampling fraction really comes out as an offset term, but now it's not a constant. It involves random variables. That's why this problem has become so hard. So I'm not going to go through some of this. I don't know what I was thinking, but I will just give you some references in terms of the code and the selection bias work that we did here. But I want to talk about this COVID-19 research that we did. Since we were sort of like, really set out that we can do COVID, that we can correct the CHR data, we were able to take the same type of association models that we showed you, right? Logistic model. But now the outcome is a COVID outcome. The risk factors are what we are interested in. Is there racial disparities? Is there a pre-existing comorbidities? And so we were able to do this because all the machinery was already set. So we were able to really, were the first few papers which showed that in Michigan, there's a strong racial disparity in terms of COVID-19 outcomes, and that continued. And what we showed is that, yes, outcomes have really improved, but Blacks were still at much higher relative risk of COVID-related severities, which is a phenomenon that is known as the color of COVID. Things have improved over time, but not on a relative scale in the same rate. That's very important to know for understanding health disparities. We were also doing this phenome-wise agnostic studies of really looking at what puts them at high risk factors. And now many of these is known right now, but we are really giving this information to the physicians real time, that if you had prior pneumonia or respiratory diseases or renal diseases, you are much higher risk of hospitalization. So, and how does it differ across Black and white ethnicities? So right now we are doing a lot of research on long COVID in terms of really setting things up right. How do you do booster studies? How do you do missing data imputation? So I just want to say that this is really, working with EHR is a lot of fun. It's fascinating, it's frustrating, but it's really worth the trouble. And I do feel that statisticians in particular have a lot to contribute to this area, because we understand the machine learning, but we also understand sampling and study design. That's really quintessential to this. I just want to say that I am a data dreamer, but I'm not the only one. You're all here. And I want to really thank the Purdue Statistics for this invitation. Sorry, I did not time my talk and I did not realize that the class is coming. But I want to thank Dennis. I want to thank Men. And in particular, I want to thank Holly for like really being immaculate with the organization of this trip. And finally, I want to thank each and every participant of Michigan Genomics Initiative who make this research possible. Thank you very much.
Well, we're about out of time, but let's think for one more time for 30 students in 692. There's a sign-in sheet here that you can sign in. If you're on Zoom, then you're all set.